Lake Forest, California. Acute binder Lisa Microils 2.0 prolactin hormone (PRL) test system product code 725-300 1.0 introduction intended use. The quantitative determination of prolactin hormone concentration in human serum by a microplate enzyme immunoassay. Colorimetric summary and explanation of the test. Isn't prolactin hormone (PRL) secreted from the lactatrice of the anterior pituitary, is a protein consisting of a single polypeptide chain containing approximately 200 amino acids. The primary biological action of the hormone is on the mammary gland, where growth of the gland and in the induction and maintenance of milk production. There is evidence to suggest that prolactin may be involved in steroidogenesis in the gonad, acting synergistically with luteinizing hormone (LH). High levels of prolactin appear to inhibit steroidogenesis as well as inhibiting LH and follicle-stimulating hormone (FSH) synthesis at the pituitary gland. 1.2 The clinical usefulness of the measurement of prolactin hormone (PRL) in ascertaining the diagnosis of hyperprolactinemia and for the subsequent monitoring the effectiveness of the treatment has been well established. 3.4 In this method, PRL calibrator, patient specimen or control is first added to a streptavidin-coated well. Biotinylated monoclonal and enzyme-labeled antibodies directed against distinct and different epitopes of PRL are added and the reactants mixed. Reaction between the various PRL antibodies and native PRL forms a sandwich complex that binds with the streptavidin coated to the well. After the completion of the required incubation period, the enzyme, prolactin hormone antibody-bound conjugate is separated from the unbound enzyme, prolactin hormone conjugate by aspiration or decantation. The activity of the enzyme present on the surface of the well is quantitated by reaction with a suitable substrate to produce color. The employment of several serum references of known prolactin hormone levels permits the construction of a dose-response curve of activity and concentration. From comparison to the dose-response curve, an unknown specimen's activity can be correlated with prolactin hormone concentration. 3.0 Principal Immunoenzymometric Assay Type 3 The essential reagents required for an immunoenzymometric assay include high-affinity and specificity antibodies, enzyme-labeled and immobilized, with different and distinct epitope recognition, in excess, and native antigen. In this procedure, the immobilization takes place during the assay at the surface of the microplate well through the interaction of streptavidin coated on the well and exogenously added biotinylated monoclonal anti-PRL antibody. Upon mixing monoclonal biotinylated antibody, the enzyme-labeled antibody and a serum containing the native antigen, reaction results between the native antigen and the antibodies, without competition or steric hindrance, to form a soluble sandwich complex. The interaction is illustrated by the following equation. Simultaneously, the complex is deposited to the well through the high-affinity reaction of streptavidin and biotinylated antibody. This interaction is illustrated below. Ag-PRL BTN AB M antigen, antibodies sandwich materials provided, APRL calibrators -1 ml vial 6 BTN AB M plus streptavidin.W. Immobilized streptavidin CW equals streptavidin immobilized on well immobilized complex equals sandwich complex bound to the well after equilibrium is attained. The antibody bound fraction is separated from unbound antigen by decantation or aspiration. The enzyme activity in the antibody bound fraction is directly proportional to the native antigen concentration. By utilizing several different serum references of known antigen values, a dose-response curve can be generated from which the antigen concentration of an unknown can be ascertained. 4.0 Reagents 4.0 Reagents materials provided, APRL calibrators -1 ml vial icons A, F6 vials of references for PRL antigen in human serum at levels of 0, A, 5, B, 10, C, 25, D, 50, E, and 100, F, and G/milliliters. Store at 2-8 degrees C. A preservative has been added. Note: the calibrators, human serum-based, were calibrated using a reference preparation, which was assayed against the WHO third is 84/500th. BPRL enzyme reagent -13 ml/vial, Icon 1. 1. Vial containing enzyme labeled antibody, biotinylated monoclonal mouse agent buffer, dye, and preservative. Store at 2-8 degrees C. C-reactivity coated plate -96 wells. Icon 196-well microplate coated with streptavidin and packaged in an aluminum bag with a drying agent. Store at 2-8 degrees C. Dewash solution concentrate -20 milliliters. Icon greater than 1. 1. Vial containing a surfactant and buffered saline. A preservative has been added. Store at 2-8 degrees C. E-substrate a -7 milliliters / vial. Icon SA1. 1. Vial containing tetramethylbenzidine, TMB, in buffer. Store at 2-8 degrees C. F substrate B -7 milliliters / vial. Icon SB1. 1. Vial containing hydrogen peroxide, H2O2, in buffer. Store at 2-8 degrees C. Stop G-stop solution -8 milliliters / vial. Icon 1. 1. Vial containing a strong acid, 1N-C. Store at 2-8 degrees C. H product instructions, Note 1. Do not use reagents B1 the kit expiration 8. Note 2. Avoid extended exposure to heat and light. Opened reagents are stable for 60 days when stored at 2-8 degrees C. Kit and component stability are identified on the Note 3. Above reagents are for a single 96-well microplate. 4.1 Required but not provided. 1. Pipette capable of delivering 0.025 and 0.050 milliliters, 25 and 50 microsinal volumes with a precision of better than 1.5%. 2. Dispenser S for repetitive deliveries of 0.100 and 0.350 milliliters, 100 and 350 ml volumes with a precision of better than 1.5%. 3. Microplate washers or a squeeze bottle optional. 4. Microplate reader with 450 and 620 nm filters. 5. Absorbent paper for blotting the microplate well. 6. Plastic wrap or microplate cover for incubation steps. 7. Vacuum aspirator optional for wash steps. 8. Timer. 9. Quality control materials. 5.0. Precautions for in vitro diagnostic use, not for internal or external use in humans or animals. All products that contain human serum have been found to be non-reactive for hepatitis B surface antigen, HIV 1 and 2, and HCV antibodies by FDA-licensed reagents. Since no known test can offer complete assurance that infectious agents are absent, all human serum products should be handled as potentially hazardous and capable of transmitting disease. Good laboratory procedures for handling blood products can be found in the Center for Disease Control National Institute of Health, Biosafety and Microbiological and Biomedical Laboratories, 2nd edition, 1988, HHS, publication No. CDC, 88-8395. Safe disposal of kit components must be according to local regulatory and statutory requirement. 6. Specimen collection and preparation. The specimen shall be blood, serum in type, and the usual precautions in the collection of venipuncture samples should be observed. For accurate comparison to established normal values, a fasting morning serum sample should be obtained. The blood should be collected in a plain retop venipuncture tube without additives or anticoagulants. Allow the blood to clot. Centrifuge the specimen to separate the serum from the cells. In patients receiving therapy with high biotin doses, E. greater than m/day, no sample should be taken until at least 8 hours after the last blood in administration, preferably overnight to ensure fasting sample. 5 days. If the specimen S cannot be assayed within, the samples may be refrigerated at 2-8 degrees C for a maximum period of time. The sample S may be stored at temperatures of -20 degrees C for up to 30 days. Avoid use of contaminated devices. Avoid repetitive freezing and thawing. When assayed in duplicate, 0.050 milliliters, 50 ml of the specimen is required. 7.0 Quality control. Each laboratory should assay controls at levels in the low, normal and elevated range for monitoring assay performance. These controls should be treated as unknowns and values determined in every test procedure performed. Quality control charts should be maintained to follow the performance of the supplied reagents. Pertinent statistical methods should be employed to ascertain trends. Significant deviation from established performance can change in experimental conditions or indicate a noticed degradation of kit reagents. Fresh reagents should be used to determine the reason for the variations. 8.0 Reagent preparation. 1. Wash buffer. Dilute contents of wash concentrate to 1000 milliliters with distilled or deionized water in a suitable storage container. Store at room temperature 20-27 degrees C for up to 60 days. 2. Working substrate solution, stable for 1 year. Pour the contents of the amber vial labeled solution into the clear vial labeled solution B. Place the yellow cap on the clear vial for easy identification. Mix and label accordingly. Store at 28 degrees C. Note 1. Do not use the working substrate if it looks blue. Note 2. Do not use reagents that are contaminated or have bacteria growth. 9.0 Test procedure. Before proceeding with the assay, bring all reagents, serum reference calibrators, and controls to room temperature 20-27 degrees C. Test procedure should be performed by a skilled individual or trained professional. 1. Format the microplate wells for each serum reference calibrator, control and patient specimen to be assayed in duplicate. Replace any unused microwool strips back into the aluminum bag, seal and store at 2-8 degrees C. 2. Pipette 0.025 milliliters, 25 microsynal of the appropriate serum reference calibrator, control or specimen into the assigned well. 3. Add 0.100 milliliters, 100 ml of PRL enzyme reagent solution to all wells. 4. Swirl the microplate gently for 20-30 seconds to mix and cover. 5. Incubate 60 minutes at room temperature. 6. Discard the contents of the microplate by decantation or aspiration. If decanting, blot the plate dry with absorbent paper. 2. Add 0.350 milliliters, 350 ml of wash buffer. See reagent preparation section. Decant, tap and blot, or aspirate. Repeat 2.2 additional times for a total of 3.3 washes. An automatic or manual plate washer can be used. Follow the manufacturer's instruction for proper usage. If a squeeze bottle is employed, fill each well by depressing the container, avoiding air bubbles, to dispense the wash. Decant the wash and repeat 2 additional times. 7. Add 0.100 milliliters, 100 ml of working substrate solution to all wells. See reagent preparation section. Always add reagents in the same order to minimize reaction time differences between wells. Do not shake plate after substrate addition. 8. Incubate at room temperature for 15 minutes. 9. Add 0.050 milliliters, 50 ml of stop solution to each well and gently mix for 15-20 seconds. Always add reagents in the same order to minimize reaction time differences between wells. 10. Read the absorbance in each well at 450 nm using a reference wavelength of 620-630 nm to minimize well imperfections in a microplate reader. The results should be read within 30 minutes of adding the stop solution. 10.0 Calculation of results. A dose-response curve is used to ascertain the concentration of prolactin hormone, PRL, in unknown specimens. 1. Record the absorbance obtained from the printout of the microplate reader as outlined in example 1. 2. Plot the absorbance for each duplicate serum reference versus the corresponding PRL concentration in ng/milliliters on linear graph paper. Do not average the duplicates of the serum references before plotting. 3. Draw the best fit curve through the plotted points. 4. To determine the concentration of PRL for an unknown, locate the average absorbance of the duplicates for each unknown on the vertical axis of the graph, find the intersecting point on the curve, and read the concentration in mg/milliliters. From the horizontal axis of the graph, the duplicates of the unknown may be averaged as indicated. In the following example, the average absorbance 1.374 intersects the dose-response curve at 36.1 mg/milliliters, PRL concentration, see Figure 1. Note: computer data reduction software designed for ELISA assays may also be used for the data reduction. If such software is utilized, the validation of the software should be ascertained.